Okay, well, let's look at the relationship between DNA, which we see here, our double helix, which has our phosphates and sugar sides. It happens to be deoxyribose sugar. We see our nitrogenous bases here. As we said before, adenine always goes with thymine, cytosine always goes with guanine. So those are called obligatory base pairings or complementary base pairings. And what we see is, of course, DNA. Importance of DNA is that the major importance is that it actually contains genes, and what genes are, they are instructions of how to build proteins, because our cells are primarily made out of their organic molecules, and proteins are the most important thing. So how are we going to do that? Well, what we want to look at in this particular picture is the relationship between DNA and chromosomes. Now, first of all, a lot of people don't realize this, but your DNA is not found as chromosomes all the time, but only wind up into chromosomes whenever a cell's getting ready for our division, our mitosis that we talked about. Now, so DNA is usually found in most cells that are not dividing when they're in the interphase stage in unwound DNA or unwound chromosomes, and we call that chromatin. So DNA is usually found in the form of chromatin inside of our nucleus. But if a cell does need to divide, of course, what we've got to do is we've got to duplicate the DNA. Therefore, we have to actually make what we call sister chromatids, which are duplicates of each other. So when the cell divides, we can pull one chromatid to one side, the other chromatid to the other side, and that assures that our new cells have all the DNA that they need. So, but what we're looking at here is we're looking at the process where DNA actually wraps around proteins called histones. They form large complexes called nucleosomes, which then further wrap into the actual sister chromatid, which is duplicated and connected to another sister chromatid with a centromere. So the centromere, again, is the connective area between the two sister chromatids. And of course, this forms what we typically think of as the typical looking kind of X-shaped chromosome. So again, when we talk about this, we need to understand that DNA is usually unwound in the form of chromatin. Now, what we're going to look at is the process which is called protein synthesis, made up of two steps, transcription and translation. So we're going to look at the process by which we take the gene on the chromosome and we get a protein from it. So that's what we'll talk about next.